So thanks to those of you who have been here all 3 weeks. This is the third week talking about the evidence bearing on intelligent design and biological evolution. Today, in the previous session, we talked about essentially the introduction to intelligent design, the basic principles lying behind it. The second session, we talked about a few of the scientific challenges to evolutionary theory. And today I want to continue that discussion by talking about some of the challenges to naturalistic origins of life scenarios, in particular in relation to two main challenges that I'm going to talk about today. The first of those being the origins of protein folds, and the second being instances of causal circularity. I'll talk about what those are when we get there. So let's first of all talk about the origins of protein folds. So for those of you who have a scientific background, you'll have to excuse me. I need to give I need to give a basic introduction to protein structure in order to communicate this argument. So just bear with me while I go through some of the basics here. So we have so the primary structure of proteins is made up of the amino acid sequence. Amino acids are the building blocks or the subunits of proteins, and the sequential arrangement of those amino acid subunits by virtue of the properties of the side chains of those amino acids determines how a protein will fold into a three-dimensional structure. So the secondary level of structure of proteins are these alpha helices and beta sheets that you see here, and then these pack together to form what's known as the tertiary structure of proteins, which you can see here, and these form basically domains of proteins. And then you have quaternary structure, which is the next level of protein organization, where you have multi-domain proteins, where these domains pack together to form these quaternary structures. Amino acids, as I said, amino acids are the subunits, the building blocks of proteins, and these are linked together by peptide bonds. So you can see these are some of a couple of the amino acids, glycine and alanine, and their chemical composition are the same, except for the properties of the side chains, which are also known as the functional R groups of these amino acids. And they have the amino acids all have two ends. They have what's called the amino group, or the N terminus, and they also have the carboxyl group, or the C terminus. And these link together, they give off water. It's known as a condensation reaction to form a peptide bond. And this is glycylalanine. So glycine and alanine have linked together, and there's a peptide bond formed in the middle. And so that's how proteins are. What proteins are comprised of. These amino acid chains are linked together, and then the properties of the amino acid side chains determines how the protein collapses into a three-dimensional structure. So here we can see the backbone of the polypeptide, and you can see different side chains that come off from these different amino acids, and it's the properties of those side chains that give the protein its structure. There are 20 different amino acids that are commonly found in proteins, as you can see here, and these have different properties. So some are negatively charged, some are positively charged, some are polar, and some are non-polar. So polar just means it's hydrophilic, and non-polar means it's hydrophobic. And basically the protein will fold. There'll be various bonds that help the protein to fold into its three-dimensional structure, and any single one of these bonds is weak by itself, but then many of them together create a strong bonding arrangement that stabilizes the particular three-dimensional structure, as you can see here. And hydrophobic forces help proteins to fold into compact conformations. The polar amino acids, side chains tend to be displayed on the surface. So these are hydrophilic amino acids, and they can interact with water. And the non-polar amino acid side chains, which the hydrophobic ones, are buried on the inside to form this tightly packed hydrophobic core of atoms that are hidden from water. Okay, so that's just a little bit of the basics of protein folding. And hydrogen bonds within a protein molecule help to stabilize its folded shape, as you can see here. And there's different hydrogen bonds within that protein molecule that stabilize its structure. Proteins come in a wide variety of shapes and sizes. So there's thousands of different protein structures. Here's just a sample of some of them. Some of those might be familiar to you, like insulin, for example. or cytochrome C, etc. Hemoglobin, which carries oxygen around in the blood, and so forth. Okay, so having given some of that background of protein structure, I want to talk about the evolvability of protein folds. And remember that proteins are the building blocks of molecular machines that we have in the cell. They also function in biochemical pathways, converting substrates into products, and they basically run the show, govern the show in biology. Now, how can we evolve protein structures? Well, what do we need to look at to assess the evolvability of these protein structures? Well, as I said before, there are 20 different amino acids that are used in cellular proteins. And so that means that for a modestly length protein of 150 amino acids, there are 20 raised to the power of 150 possible combinations, right? So that's 20 times 20 times 20 times 20, all the way up to 150. So that's a huge combinatorial space. To put that in context, there's only, it's thought by physicists that there are approximately 10 to the 80 atoms in the observable universe. So the combinatorial space of amino acids of a relatively modestly length residue of 150 amino acids, that is beyond the number of atoms in the observable universe. So the question that we can ask is, how prevalent are functional protein folds in combinatorial space? But we know that many of those sequences of amino acids form stable and functional protein structures, but we also know that many of them don't. Many of them are gibberish. And so the question that we can ask is, what is the ratio of stable and functional protein structures within that vast array of combinatorial possibilities? Just as in the English language, sentences and paragraphs convey meaning because there's far more ways of writing gibberish than there are ways of writing functional sentences. It's functional stringing alphabetic characters together from meaningful sentences and paragraphs and so forth. And so the question that we can ask is the same thing true in protein structures? If it turns out that the relative prevalence of functional protein folds is astronomically small, then it seems to me that it would be very difficult by a blind, unguided search to find those functional and stable protein structures among the vast number of alternatives. Whereas, on the other hand, if it turned out that it was relatively abundant, that you would have a relative abundance of functional protein folds within that vast array of combinatorial possibilities, then it would seem much more reasonable to expect that a blind, unguided search could, by chance, find those functional protein structures that could then be selected on by natural selection. So there's a number of papers that have been published on this. Probably the most well-known or the most cited on this is Dr. Douglas Axe's work in 2004 that was published in the Journal of Molecular Biology. At the time, he was a postdoc at Cambridge University in the UK. But I want to talk about another paper, a more recent paper, which also I think bears strongly on this question, perhaps a less well-known or less cited paper in discussions of this. And that is this paper that was published in Nature it's called Robustness Epistasis Link Shapes the Fitness Landscape of a Randomly Drifting Protein. And basically after, so in this paper, so they basically conducted a mutagenesis experiment where they induced mutations into a particular protein structure. And according to the paper, after only a few random mutations under weak selection, around a third of subsequent changes to a protein completely disabled it. And after several more mutations accumulate, the protein is inactivated by slightly under 2/3 of subsequent changes. And after random alteration of less than 10% of the protein's initial sequence, it becomes permanently non-functional. So the fitness approach is 0. So how can the results of this paper help us to elucidate the relative rarity or abundance of stable and functional protein structures within that combinatorial space? Well, the corresponding rarity can be calculated by working backwards, right? So the number of sequences that differ from an optimal one by a given number of amino acids increases almost exponentially with the number. So a random search would find a barely functional sequence long before an optimized one. So estimating the upper limit for the probability of a successful trial must focus on the neighborhood of sequence space. So let's do the math. So in the study, after 5 to 10 mutations, roughly 2 in 3 mutations inactivate a protein. Therefore, 1 in 3 amino acids at each position on average would correspond to a functional sequence. The rarity would then be less than a third to the power of the sequence length. And this estimate closely matches the result of Axe's 2004 experiment on beta-lactamase, that only 1 in 10 to the 77 sequences corresponds to a functional fold or domain within the protein. And the actual rarity is actually much more extreme, since almost no sequences are functional after 10% of a protein randomly changes. So this was the previous paper by Dr. Douglas Axe, which is very well known on this. The experimental procedure gets a little technical, so if anyone wants the technical details, I'm happy to correspond with them or talk to them afterwards. But basically, he directed a site-directed mutagenesis experiment where he randomly altered the amino acid sequence of a particular domain of beta-lactamase, and according to his paper, his estimated ratio of stable and functional protein structures within combinatorial space is roughly 1 in 10 to the 74th power. And so to put that in context, again, there's only 10 to the 80 subatomic particles in the known universe and approximately 10 to the 65 atoms in the galaxy. So an astronomical rarity of these functional folds within that combinatorial space, the number of ways there are of arranging the amino acids. This presents a probabilistic resource problem. So there are approximately 10 to the 80, as I said, elementary particles in the observable universe since the Big Bang. Roughly 10 to the 16 seconds have elapsed. And the time it takes light to traverse the smallest distance, or the Planck length of 10 to the minus 33 centimeters, is approximately 10 to the minus 43 seconds. So by multiplying together the 3 relevant factors of 10 to the 80 times 10 to the 16th times 10 to the 43, we can determine that the maximum number of events that could have taken place in the observable universe since its origin is 10 raised to the 139th power. We can call that the universal probability bound. So the challenge of these papers, and there are others as well that I haven't discussed, is that locating the functional sequences within the vast sea of combinatorial possibilities by a random search is like finding a tiny needle in an enormous haystack within a very limited time. I'm going to show a short video where Doug Axe and his colleague Stephen Meyer talk about this research.
Richard Dawkins, the famous Oxford evolutionary biologist, has illustrated how the Darwinian mechanism works using a metaphor he calls climbing Mount Improbable.
From the front side, the mountain is a sheer cliff that could never be scaled in one giant leap. For Dawkins, this represents the impossibility of creating a complex animal by chance alone. Yet Dawkins also envisioned an alternative route up the backside of Mount Improbable, a long, gradually sloping trail of small steps leading all the way to the summit.
According to Dawkins, that's how you'd climb the mountain. And that's also how you'd build a Cambrian animal, one small step at a time. What chance alone can't accomplish in one blind leap, natural selection can accomplish through the cumulative effect of many small incremental steps.
In theory, each step corresponds to a small unit of biological change, a new gene and its protein product. But do mutations and natural selection have a reasonable chance of producing even one protein in the time available? Since 1992, molecular biologist Doug Axe has examined this question.
There's a story that's being told, and there's a appeal in the case of Darwinism to random mutation and natural selection as being, in vague terms, the mechanism. But if you look at the detail, what kind of mutation can accomplish these transitions? And there, it's important to realize that the one area where we can really nail this down is at the single protein level, where you can actually measure it. And if you look at protein structures, to get a substantially new protein fold is prohibitively difficult.
Each of the thousands of different proteins in nature is actually a chain made from a specific combination of 20 different amino acids. The sequential order of these chemical building blocks is crucial, for if they are arranged correctly, the chain folds into a functioning three-dimensional molecule. But if the amino acids are incorrectly assembled, no protein will form. If proteins are indeed rare among the possible sequences of amino acids, what are the odds that mutations would stumble upon a functional combination of chemicals from the vast number of alternatives? To find out, Axe randomly altered the structure of an enzyme protein comprised of 150 amino acids.
You've got a protein 150 amino acids long, then you've got 20 to the 150th power possible ways of arranging the amino acids. Out of all those possibilities, how many are functional and how many are gibberish?
If you do the experiments and you analyze how much information is required to get, say, a new protein fold, is just far beyond what you can get by random mutation and natural selection.
How far beyond? Axe published his findings in the Journal of Molecular Biology. He determined that among all the possible amino acid combinations, the probability of generating just one short protein by mutation is roughly 1 in 10 to the 74th power, or 1 chance in 100 trillion. Trillion.
To put that in context, there's only 10 to the 65 atoms in the entire galaxy. So to build a new functional protein by selection and mutation within the time allowed for the Cambrian explosion, what you're essentially having to do is equivalent to a blindfolded man looking throughout the entire galaxy for one marked atom. So what we're talking about is searching for a tiny, tiny needle in an enormous haystack and having a very limited time to search.
So on the question of something like the Cambrian explosion, there does not appear to be any way that unguided random mutations can accomplish what needs to be accomplished to explain new functional proteins. And certainly by extension, wherever in the history of life you would need to have multiple new protein folds, the probabilities multiply. So there's no reason to think that this is plausible.
Okay, so the problem, though, extends deeper. When molecular machines require multiple cooperating proteins in order to perform their function, the probabilities multiply exponentially. So here we see a diagram of the replisome, the replication machinery that replicates the DNA to prepare for cell division. And as we talked about in the first session, the DNA replosome or replication machinery requires a myriad of different functional protein components that have to be specifically crafted in order to fulfill their respective roles of synthesizing DNA. So the probabilities then multiply exponentially as we deal with these molecular machines that require a whole concert or a myriad of multiple protein components that have to be specifically crafted to fulfill their respective jobs in order to accomplish a high-level objective, such as replicating DNA. Now, as you might have gathered or suspected, evolutionary biologists have put forward some attempts at responding to this sort of argument. And one of those objections that sometimes put forward is that maybe the sequences are rare, but not isolated. So the idea here would be that that although they're astronomically rare, they're clustered together in sequence space, so that you could hop from one island of function to another relatively easily. So to illustrate why I think this doesn't solve the problem, consider this sentence in the English language: no biological information by natural means. So this text is, you might call it, linguistically isolated. Now that doesn't mean that it can't be altered and still remain readable, or that it can't or it cannot be altered and remain meaningful. What we're saying is that there is no freedom, essentially, to generate new meaningful phrases from this sentence if we restrict ourselves to only single character changes, and we require a meaningful phrase at every step. So we can't take this sentence and cause it to undergo open-ended evolution over time. So we might be able to get, we can get to perhaps no biological information by natural meats. We might change the means to meats or something like that, which would still be meaningful, although it is not particularly coherent sentence. It still retains its meaning at the word level, at the level of individual words, but it loses all coherence as a phrase. So we're stuck on an isolated island with just a population of two sentences, and there's not especially much difference between those two meanings. Now, if we take something much smaller, the transitions can keep going. So if we take just a 3-letter word in the English language, red, we have rod, and then mod, and mad, and mat, and cat, and car, and tar, and bar, and ban, and so forth. And so we can just keep changing one letter at a time and cause it to retain functional significance. Now, there's approximately 3 or 4% of possible 3-letter combinations are actually English words. And so that makes 3-letter words rare in the sense that they are outnumbered by nonsense combinations of 3 letters, and yet they don't seem to be isolated. And so although they're rare, they're clustered together in sequence space. But it seems that the longer your sentence, the more not just rare, but also isolated in sequence space your sequences are going to be. So there are 25 ways to change a given letter into some other letter. And so that means that there are 75 ways to mutate any 3-letter word with a single letter change. And so even if most of these mutants are nonsense, a success rate of 3 to 4% means that 2 or 3 of those words are expected on average to be English words that we recognize. And that is also bolstered by the fact that there are certain patterns of vowels and consonants that are more word rich than others. So if we go back to the first phrase that we considered, no biological information by natural means, there are about 1,000 ways to make a single letter change to it. So there's 42 different characters, 42 different positions, and there's 26 letters of the alphabet, so 26 alternatives to each position. So that's approximately 1,092. That's approximately 1,092 possible mutants, including spaces, but not punctuation. So we might expect to avoid isolation of meaningful phrases among all 42 combinations are more prevalent than about 1 in 1,000. And so if meaningful phrases are much less common than that, then we would expect there to be isolation of sequences, or functional sequences, or meaningful sequences. And it turns out that meaningful 42 character combinations are far more rare than 1 in 1,000, which explains why the islands of meaning are extremely isolated. They're very isolated in a vast sea of combinatorial possibilities. Now, of course, there are many ways that you can arrange 42 characters to spell out sentences that have meaning. And almost all of these can be expressed in different ways, or said in different ways, stated in different ways. But that doesn't amount to anything compared to the vast sea of combinatorial combinations of that length, which is 27 raised to the 42nd power, or 10 raised to the 60th power. So there is a relationship, I think, between rarity and isolation here. Now, another objection that sometimes comes up. So when we're the fact the rarity and isolation of stable and functional protein structures within combinatorial space is not merely a problem for the origins of life, but it's also, I would argue, a problem for biological evolution more broadly than that as well. Getting from one island of function to another by a blind, unguided search. Now, one scenario that's often put forward by evolutionary biologists is this idea of domain shuffling or exon shuffling. And this is the hypothesis that fundamentally new protein folds can be created by recombining already existing domains. And that's thought to be accomplished by moving exons, those are parts of genes that code for proteins, from one part of the genome to another. And this is known as exon shuffling. And there are several ways by which exon shuffling can occur. The most common are probably mediated by transposons, which are jumping genes, genes that can move from one part of the DNA to another, and also crossover during meiosis, which is replication of sex cells, and recombination. So so exon shuffling then can be transposon mediated, or it can occur as a result of crossover during meiosis and recombination between non-homologous or sometimes, but less frequently, short homologous DNA sequences. And another process known as alternative splicing which is the way that RNA is processed, such that exons are mixed and matched in different configurations in order to express different proteins, is also thought to play a role in facilitating this exon shuffling. Now, if you want more detail on some of the problems with this, I've written an article series of 2 articles, which I'm happy to send if anyone's interested. But let me just summarize a couple of problems with this scenario. One is that the model at its core presupposes the prior existence of protein domains. Secondly, the model assumes the ability of blind evolutionary processes to account for a specific toolbox of domains that can be recombined in various ways to yield new functions. And thirdly, the domain shuffling hypothesis in many cases requires a formation of new binding interfaces, for interface compatibility. So when it comes to the origins of life, the problem though only gets worse. And this is because each of the amino acids comes in 2 different isomeric forms, or 2 different flavors. You can have the left-handed isomeric form of amino acids, and you can also have the right-handed form of amino acids. And when it comes to biological proteins that we find in our bodies, these are uniformly of the left-handed isomeric form. Whereas when you have experiments that are conducted that produce sets of amino acids, such as the famous 1953 Harold Urey and Stanley Miller experiment, what they were able to produce was what's known as a racemic mixture of amino acids, where you have approximately a 50-50 mixture of left-handed and right-handed isomeric forms. And that's no good. You need all of one-handed isomeric form. So that multiplies those improbabilities yet further, because at each site, if you have a 150 amino acid long residue, then each site you have a 1 in 2 chance at the origins of life of getting a left-handed versus a right-handed isomeric form. Now, as it might not surprise you to learn that also origins of life theorists know about this problem and have offered possible solutions to it. This is a review paper that was published in 2010. And the lead author of this paper had previously done his own research in 2004. He's leads a Japanese team. And they reported that they had performed recrystallizations using a mixture of 12 amino acids with an excess of asparagine. And that was those amino acids were in a racemic mixture, so a 50-50 split of left-handed and right-handed forms. And the result was that all of the amino acids with the same configuration as the asparagine, one of the amino acids, were co-crystallized preferentially. And the authors took this to suggest that it's the nature of a mixture of racemic amino acids to produce a spontaneous high enantiomeric excess. So in other words, they argued, the evidence showed that homochirality could in fact be achieved. And the lead author of this paper published this subsequent review paper in which he surveyed the evidence supporting his thesis that chiral enhancement in one amino acid can result in chiral enhancement in other ones following recrystallization. Now, the results from this study are certainly very interesting, and I would argue that it's at best only a small step towards resolving the problem of achieving homochirality. So asparagine was not one of the amino acids that was actually found in the Stanley Miller experiment, and is also unlikely to have been present on the early Earth in significant quantities. So whatever excess there was would most likely be washed away by the next rain and remixed. So it's an interesting study for sure, but I'm not convinced that it solves the problem entirely. So let me talk about one more problem facing naturalistic origins of life scenarios before we conclude and open up to questions. And that is the problem of causal circularity. And I'm just going to give a couple examples of this. So causal circularity are essentially chicken and egg problems. So let me give a basic overview of the central dogma of molecular biology, which is necessary to understanding the first of those examples. So as most of you probably know, you've got the DNA here. It's transcribed using an enzyme known as RNA polymerase into messenger RNA, which is a single-stranded intermediary molecule. And in a eukaryotic cell, it would be taken outside the nucleus to the ribosome, which is a two-part chemical factory that is the site of protein synthesis, and there it would be translated into a polypeptide. And then that polypeptide would fold into a three-dimensional protein structure and carry out some function in the body, such as might be a protein involved in cell division, or wing shape, or code for antibody resistance, or something like that. So here's a short animation that shows the process of information flow in the cell.
The DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene. At this point, the RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons. Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay, so and a series of enzymes catalyzes a reaction that catalyzes a series of enzyme catalyzed reactions forms a linked pathway. So each of the amino acids that we see in that we see contribute to these proteins, and indeed all of the nucleotides, have their own biosynthetic pathway where you go from molecule A, that's catalyzed by an enzyme into molecule B, and that's now will be catalyzed by another enzyme into molecule C, and then so forth, until eventually you have your product, which will be one of the nucleotides or one of the amino acids that we use for proteins and for the DNA and the RNA. So this presents a causal circularity problem. So each amino acid and nucleotide has its own biosynthetic pathway. And each step of a biochemical pathway is catalyzed by an enzyme. And enzymes are composed of amino acid subunits, which are coded for by the sequence of nucleotides in the DNA. So this presents a causal circularity problem where the enzymes are themselves made up of amino acids, which are and yet they are involved in the biosynthetic pathway to production of the amino acids. And likewise, the sequence of amino acids in those enzymes is specified by the sequential arrangement of the nucleotides in the DNA, but each of the nucleotides that is found in the DNA has its own biosynthetic pathway which is catalyzed by those enzymes. So that's a causal circularity problem, or a chicken and egg problem. A related causal circularity problem is, which is very well known, is that DNA codes for proteins, and yet proteins replicate and transcribe the DNA. So the question is, which came first, DNA or protein? Because the DNA needs the protein to replicate it and to transcribe it into messenger RNA, and there's also protein involved in translation of the messenger RNA into protein. There's the amino acyltransferase synthetases, for example, which glue the incoming amino acids to the transfer RNA molecules, and there's a protein component of the ribosome, and so forth. So which came first, DNA or protein? DNA needs proteins, and proteins are coded for by DNA. So origins of life researchers are, of course, again, very aware of this problem, and so they put forward what's known as the RNA world scenario, which is to say that the first genetic material was actually not DNA, but actually RNA. And the basis for that is the RNA is able to serve as an information storage medium, and it also is able to fold into three-dimensional structures to perform some limited catalytic roles. And so the idea is that in regards to the question which came first, the chicken or the egg, well, at some point the chicken actually was the egg, because the RNA is able to perform both the function of DNA as well as the function of protein. So that is thought to have been the predecessor to modern cells, where you first go from the RNA-based system, and then you have evolution of RNAs that can direct protein synthesis. So you have RNAs coding for protein, and then finally you have DNA taking over as a genetic material, and RNA becoming an intermediate molecule between DNA and protein, which is the situation that we currently observe in the cell. Now, there's a number of problems with the RNA world scenario. If you want more detail on this, I've written quite a number of articles, which I'm happy to send you. And also this is discussed in quite a bit of detail in Stephen Meyer's book, Signature in the Cell, as well as other books and papers as well. So happy to send resources if anyone's interested. One problem with the RNA world scenario is that RNA building blocks are hard to synthesize and easy to destroy. So at the boiling point of water, adenine and guanine possess a half-life of 1 year. These are nucleotide bases. The half-life of uracil and cytosine is 12 years and 19 days, respectively. And in hydrothermal vents, and it's the consensus among origin of life theorists that the origins of life took place in hydrothermal vents deep in the ocean, which are get to very, very high temperatures. The temperature would have been much higher than this, much higher than the boiling point of water. So it seems unlikely that RNA would have been able to survive easily on the early Earth. Furthermore, RNA is very unstable. It's single-stranded for one thing, which makes it more delicate than DNA. And also it's got an extra hydroxyl group, or OH group, at the 2' carbon position that DNA does not have, which makes it more chemically reactive as well. So RNA is less stable than DNA, and so it would be very unlikely to survive in the early Earth, certainly before the origins of the cell membrane, etc. Another problem is that ribozymes are poor substitutes for proteins. In fact, to date, scientists have shown that ribozymes can perform only a small handful of the thousands of functions performed by modern proteins. Just a few well-known functions of ribozymes. RNA molecules can cleave other RNA molecules at the phosphodiester bond through hydrolysis. RNA molecules can ligate separate strands of RNA by catalyzing the formation of phosphodiester bonds. And ribosomal RNA promotes peptide bond formation within the ribosome and can promote peptide bonding outside the ribosome, though only in association with an additional enzyme. And beyond that, RNA can perform only a few minor functional roles, and then usually as a result of scientists intentionally engineering the RNA catalysis in question. So it requires intentional engineering to allow it to perform the role that it does. Thirdly, an RNA-based translation and coding system is implausible because an RNA-based replication system eventually would have to begin to produce proteins, and not just any proteins, but proteins that are capable of template-directed protein manufacture. But if that is to occur, then the RNA replicator first would need to produce machinery for building proteins. So as a first step towards building proteins, the primitive replicator would need to produce RNA molecules that are capable of performing the functions of the modern proteins involved in translation. So there's a few reasons why I think the RNA-based translation and coding system is not particularly plausible. Fourth, the RNA world doesn't explain the origins of genetic information. So even if a system of ribozymes for building proteins had arisen from an RNA replicator, that system of molecules would still need information-rich templates for building specific proteins. So I personally don't think that the RNA world scenario really helps you to get around the causal circularity problem of DNA coding for proteins and proteins being required for replicating and transcribing the DNA molecule. So in summary then, stable and functional protein structures are astronomically rare relative to the vast sea of combinatorial possibilities for sequencing amino acids. And causal circularity problems present a formidable obstacle to naturalistic origins of life scenarios. And I'll finish with that, and happy to take your questions. Thanks for your attention.
Great. So thank you, Jonathan, for this session here. Lots of deep content here. What I'd like to do is similar to what we did last time, have a session first for more general questions, and then spend some time for those who want to stick on with a little more scientifically detailed ones. I've got a bunch of questions which I'd love to personally go through as well. So the first question here, and I'm not going to go in order. I'm going to kind of jump around a little bit here. But the first question is, thank you, Dr. McLatchie, for this enlightening lecture series. Such deep scientific exploration of origins and evolution of life, however, does make many books in the Bible look as rudimentary and allegorical. For example, the Genesis narrative, which was probably meant to be read as fact anyways. What that then subsequently does is raise questions on other books of the Bible, that is accuracy and reliability of the Gospels or cultural relevance of the epistles. As a scientist, how do you approach any such possible liberalization of the Bible, where we change our interpretation based on changes in culture and science?
Interesting. So this question seems a little bit off topic, but I'm happy to deal with it anyway. So in terms of reliability of the Gospels, so I've written extensively on that topic and done a number of debates on the topic as well in public lectures. So I direct you to those. You can find a lot about the reliability of the Gospels written about on my website, jonathanmclatchie.com. Just click on latest writing and you'll find an archive of my articles on this topic and others. So yeah, it's too detailed a topic to go into right at this moment. In terms of the accounts in Genesis 1 to 11, I personally am a staunch defender of an old Earth interpretation, both of science and the biblical narrative. I don't think that the Genesis narrative compels one to accept a young Earth account of life's of Earth's history. I think, for example, Genesis 1:1 says, in the beginning God created the heavens and the earth was without form and void, and darkness covered the surface of the deep, and the Spirit of God was hovering over the face of the waters. And then verse 3 says, and God said, let there be light. And there was light. And the first occurrence of the phrase "and God said," notice, is in verse 3. And each of the days of creation week, if you look carefully at the Genesis 1 narrative, begins with the phraseology "and God said," "and God said," "and God said." So the suggestion here is that day 1 of creation week actually begins in verse 3, not in verse 1. And so, irrespective of what you think about the age of the biosphere, the Bible is completely silent on the age of the Earth, because it doesn't tell you, because by the time you even get to the first day of creation week, the heavens and the earth have already been created. I personally subscribe to what's called an analogical days view of the days of Genesis. my view is an extension of Jack Collins's view. Jack Collins is one of the main Hebrew translator for the English Standard Version, and he's put out a number of books on Genesis. And he takes a view that the days of Genesis 1 are intended to be understood analogically to be an analogy for the 7-day work week. And I personally have argued, as an extension of Jack Collins's view, that and I'm not alone in thinking this, that the numbers in the ancient world are often used symbolically. That is very well documented. There's cases in the Bible where that's the case. For example, in Matthew's genealogy of Jesus, at the beginning of Matthew's gospel, he mentions that there are 14 generations from Abraham to David, 14 generations from David to the exile, 14 generations from the exile to Christ, and yet he has to duplicate and skip over generations to make the math work out. And so he's using the number 14 symbolically because 14 is the numerical value of the name of David. And I think something similar is plausibly going on in Genesis 1, where the number 7 is being used in a symbolic way. In fact, if you read G.K. Beale's book, The Temple and the Church's Mission, he points out numerous parallels between the Genesis account in the Bible and temple and tabernacle construction narratives, both in Scripture and also external to Scripture. And oftentimes the temple and tabernacle construction narratives are centered around the number 7. And that's an interesting motif. And so I personally suspect that the number 7 is being used symbolically there. But I've done a lot of thinking about Genesis 1 to 11. So if you want to talk about this in more detail, feel free to contact me via my website, and I'll be happy to correspond with you about it.
The next question is, could God have directed what looks like to be evolution by intelligently mutating these proteins and genes within the simpler species he created to cause them to form more complicated life forms? This would explain the fossil record going from simple to complex. And then finally, same questioner, why do you think that God took billions of years to create life forms from the simple and then to the complex, a process that is filled with death and suffering?
Yeah, that's a good question. So as to the first question, so what was the first part again?
So, yeah, the first part was like, couldn't God have just mutated, intelligently mutated the simpler organisms in order to produce the
sure. He could have done. So I think that we have to be careful to make a distinction between pattern and process. So I think there's actually some fairly good evidence for at least modest levels of common ancestry. Happy to talk about that if you want. I think that there's some fairly good evidence for at least limited levels of common ancestry. I tend to be more skeptical of the more the grander claims of universal common ancestry. But demonstrating common ancestry only demonstrates a pattern. It doesn't demonstrate a process to account for that pattern. And the proposition of universal common ancestry actually wasn't new with Darwin. It goes back to the ancient Greeks. What was new with Darwin was the idea that natural selection coupled with chance variation, of course he didn't know what mutations were at the time, could explain the appearance of design without recourse to a designer. And demonstrating the proposition of universal common descent doesn't get you that. It only shows you that we have hereditary continuity. And then that leaves open the question of whether it's a mindless, unguided, undirected process, or whether there is directionality and intentionality and design behind it. So I think that these are two independent questions that we need to address separately. And furthermore, there's often, I think there are a number of scenarios that are open to a theist like myself about how God brought about the creation of life. And I think that we have to be careful not to draw too sharp a dichotomy between special creation and universal common descent. For example, God can create, once you start to move away from gradualism and start to embrace saltationism, or that God abruptly brings new forms of life into existence, the boundary between special creation and common descent starts to become rather blurred. I mean, God could create new biological information in the zygote, the fertilized egg or the egg cell of a previous animal in order to bring about a new form of life, or he could even just, I think reasons to believe, take the view that God take a genome of a previous organism and then manipulated it in order to bring into being a new type of organism. And the evidence, in terms of the molecular evidence for common descent, would look exactly the same if God did that. So I think that's a possibility as well.
So in other words, to rephrase what you just said there, so imagine, like, let's go back to your example from last week where you talked about the whale and creatures that lived before it that were land-based mammals. And you showed in that the timescales don't work process-wise for that to be undirected mutation. However, you would be okay with, I guess nobody really knows, but you would be okay with, in this framework of God taking some antecedent creature and somehow changing it at the zygote level or germline level, who knows, and having subsequent creatures come from the template, if you will, of these previous organisms. And that would clearly not be what a Darwinist would adopt, at least a normal Darwinist would adopt. And yet it's not what a lot of people think of when they envision special creation, where sort of things just come out of nothing from all ab initio examples of creation. So something like that might have and exhibit the relics of, for example, mutations in the prior organism. And I'd like to ask you about broken genes related to this. So, you know, there's evidence, for example, in what a lot of people will point to for evidence for Darwinism is they'll say, well, look, there are organisms that, for example, have the vitamin C pathway inside of them, but it's broken, it doesn't work. And an intelligent designer, why would he make a broken pathway inside of someone that, you know, that doesn't seem to particularly make sense? However, in this framework, it could make sense where there is an antecedent organism that God still supernaturally intervenes and somehow puts in information content into that genome. And so you see the relics of prior genes and even prior viruses. You know, I talked about this last week, that you see these evidences of viral integration into genomes that can be common from one organism to the next, which a lot of people use as support for the process of Darwinism, but it might in fact be a more limited form of or some kind of combination of common descent with supernatural intelligent design. Could you comment on that?
Sure. Yeah, absolutely. I think that the argument from endogenous retroviral elements is one of the strongest, if not the strongest, argument for common descent. I do think that the creationists have done a good job responding to this argument, and the reason is because they overlook part of the argument, or they misrepresent the argument to some extent. So the argument is basically that primates have been on the scene for something like 50 to 55 million years. And during their history, they've undergone rounds of infections by retroviruses. And retroviruses are viruses that have an RNA genome, and the RNA genome basically gets integrated into the host chromosome. And it gets reverse transcribed into DNA and integrated into the host chromosome. And if it infects the germline, then it can become passed on to future descendants. And so they end up with this graveyard of these relics of these retroviral infections. And it turns out that the distribution of these relics of these retroviral infections forms a nested hierarchy, or a family tree-like structure, which is exactly what you would expect if there actually is a family tree and these organisms are descended with modification from common ancestors. And then there are three core genes associated with retroviral sequences. There are the GAG gene, the POL gene, and the ENV gene. And these genes themselves incorporate point mutations over time, as they've been incorporated into the host chromosome. And the distribution of those point mutations forms a very strikingly similar nested hierarchical distribution as the actual inserts themselves. And then furthermore, there are sequences that serve in the retrovirus promoters known as long terminal repeat sequences at the 5' and 3' ends of the retroviral RNA sequence. And those actually have to be identical at the point of integration into the host chromosome, but then they can occur mutations. And the degree of divergence between these 5' and 3' long terminal repeat sequences actually can be used as a predictor of the time since integration. And so you've got this three-tiered argument for primate common ancestry based upon the distribution of these retroviral sequences. Now, how have creationists responded to this? Well, they've typically gone down one of two routes. One of those is to say that the retroviruses are actually often turn out to be functional within the context of the host chromosome. So, for example, the syncytiotrophoblasts, which is essential for placental formation, what holds that together are a class of proteins known as syncytin, and those syncytin proteins are coded for by the envelope gene of retroviral-like sequences. And also the LTRs from the long-term repeat sequences of the retroviruses have turned out to contribute as promoters and enhancers within the context of the human genome and so forth. The problem is that it seems that those functions were acquired post-integration. And the evidence for that is that the insertions by retroviruses are characterized by a hallmark target site duplication, which is the characteristic hallmark of insertion by integrase because of the manner in which the integrase enzyme attacks the 5' and 3' phosphodiester bonds of the DNA molecule. And so these seem to be genuine inserts. And so I do think that pointing to function really undermines the argument significantly. The other route that sometimes people go is to argue that there is a preferential insertion bias where it's inserted at particular loci in the genome preferentially over others. And there does seem to be some level of preferential integration near CPG islands, for example. But it doesn't seem to be anywhere near locus specific enough to really account for this evidence. So I think that the best explanation for this particular evidence is the common ancestor hypothesis. But it would also be consistent with the hypothesis I'm putting forward, that there is evidence that even if common ancestry is true, it cannot be accomplished by an unguided, undirected process. And so it leaves open the possibility that perhaps the intelligent designer has used previous organisms as a template to bring into being new organisms, which would be indistinguishable both from special creation as well as common descent, although not in the traditional sense. Does that answer your question?
Yeah, that does. I think this is a really, really important point here that is worth spending a little bit of time on, because one of the things that we never want to be guilty of is ignoring evidence that doesn't fit our whatever we believe, right? And on both sides. And it seems like here what you've outlined is a very compelling case. I mean, I think the probabilities are just, they're so small and from so many different angles that it's hard for me to understand why there's not more credence given from the pure naturalist, that like it doesn't feel like there's any kind of credible way to explain this. Actually, I'm going to throw out something else that you didn't mention as an argument, but I think this is a fairly agreed upon number here, that the total number of organisms that have existed since the beginning, right, in standard biological thought, consensus is around 10 to the 40th. That's all the organisms. That's mammals, bacteria, reptiles, everything. And even if Doug Axe is off by many, many orders of magnitude, I mean, even if it's not 10 to the 74th, it's 10 to the 50th, or even 10 to the 40th, to get one protein there's no possible way that one could search out the space of nucleotide space or protein space, however you want to frame it, in order to develop anything like the complexity that we have. I mean, it's not even close. And you pointed out circular. There's so many arguments that can be given there that it feels like there's a dishonesty on the other side to just grapple with these. And there's just always these kind of, let's paper over that, and there's this sort of somehow hope that it works out. But as this last discussion has pointed out, there can also be a bit of denialism there when we look at evidence that doesn't quite fit the normal creationist schemes there, such as retroviruses, such as broken genes. And I wonder how much, one, there's a very small number of people that would simultaneously be able to hold on to both as you do, and it's something that I commend you for, and that you're trying to honestly grapple with truth there. But I wonder to what extent the problem that we're in, where you have these two divided camps, is that there hasn't been enough give and take on both sides to say, like, okay, I see your point here, and I'll grant you this point, but but similarly, you have to grant there's some merits here. I don't know. I feel like so often there's just not really a true honesty and a true willingness to engage. These points here.
I totally agree. I feel like atheists and Christians are often talking past each other because neither side takes the other sufficiently seriously to really listen carefully to what they're saying. And so it ends in the two camps. They just listen to their own side, and they don't really interact carefully with what the other side has to say. And it's quite unfortunate.
What I'd love to do is, I'd love to go, we normally go 10, 15 minutes over and have more scientifically specific questions here. So if you want to drop, feel free. There was some questions that were asked about RNA stability here. So let me pull up that one. There was, on the view of instability of RNA synthesis in thermal vents, could you comment on Taq polymerase in the PCR process?
So the Taq polymerase is derived from Thermus aquaticus, which is an archaeal organism, and archaea are well known for their extremophile nature. They like to occupy extreme environments typically, but I mean the polymerase is a copying protein. So I'm not quite sure what his question, how it relates to RNA stability per se.
Yeah, I'm not sure if you want to ask your question and clarify there.
And I'm not a biologist, so I'm just wondering, could this Taq polymerase just occur in nature, like in steps? Like, is it like a ecosystem to sustain RNA synthesis process?
Well,
something along that line.
Thermus aquaticus is an archaeal cell that occurs in nature. So for those of you who don't know, prokaryotes, so there's three domains of life, right? There's three major branches of life. There's eukaryotes, which include cells with nucleus, such as our cells. And then there are cells that don't have nuclei, and these are known as prokaryotes. And these themselves are divided into two subdivisions, namely bacteria and archaea. The archaeal cells are best known for inhabiting extreme environments, so extremes of acidity and temperature. And Thermus aquaticus is an archaeal cell from which we derive the Taq polymerase that we use in polymerase chain reaction for amplifying DNA in the laboratory setting. And Taq polymerase is a DNA polymerase, right? Is copying DNA into more DNA. So it doesn't have any relevance to RNA stability.
Could you comment on what are different ways that people respond to the RNA stability question? So here in Boston, there's a Nobel laureate, Jack Szostak, who some of you know him, who has tried to push deeper into this origins of life using RNA as the hypothesis there. But as anybody knows who's ever worked with RNA, even if you don't have an RNase and you just leave it out at your in an Eppendorf on your bench, it will degrade just by virtue of hypothesis.
Exactly, and that's why in the lab scientists typically make what's called cDNA, which stands for complementary DNA, where they actually reverse transcribe RNA into DNA in order to make it easier to work with in the laboratory, because RNA degrades so quickly. So RNA instability is quite widely known. In terms of what origin of life theorists respond, actually that's something I've not seen a lot of people respond to, the problem of RNA stability. I'm not sure if there is any good response to that. I'll need to check into that.
Okay. And then could you show that screen where you had a screen before that had the number of subatomic particles and the amount of time since the universe, and then you had something about light traveling very short distances. Could you bring that up?
Yeah, sure. Okay, here we go. Share my screen. Okay, here we go.
Okay, so I'm going to integrate this with the question that was asked before, but so there's 10 to the 80th elementary particles, so not atoms, 10 to the 65 atoms according to what we heard.
Well, in the 10 to 65 atoms in the galaxy. About 10, and I mean, it's not going to vary significantly because of the way exponents work, right? The number of elementary particles and atoms is not going to vary that much.
Well, I mean, yeah, right, because there's not thousands of elementary
Yeah.
particles for one atom. So the and then you say there's roughly 10 to the 16 seconds since the Big Bang. The time it takes to traverse the smallest distance, the Planck length of 10 to the minus 33 centimeters, is 10 to the negative 43 seconds. And then could you go to the next slide?
Yeah.
So did you just multiply all these?
Yeah, correct.
And then you determine the maximum number of events. So what is the light? I wasn't sure what the point of the light.
So yeah, so that's the maximum number of events that could have taken place in the observable universe since its inception, right? So so we're trying to calculate rather the number of events that could have taken place in the origins since the origins of the universe. So we're looking at, okay, so how many elementary particles are there in the universe? How many seconds have elapsed, and how many light traversing the smallest distance is the quickest event that can take place. So that multiplying those factors together gives you the maximal number of events that could have taken place. Does that make sense?
Yeah, it seems like this is, so it's not, I don't know, it didn't strike me that this was as relevant for questions about life on Earth, because I mean, this is,
yeah,
you're
it's trying
to be so generous here, I think maybe that it becomes almost too generous and partly irrelevant. I don't know, that was the,
yeah, it's generous, but when it comes to origins of protein structures, as soon as you are trying to build a multi-protein machine, such as the DNA replosome or something like that, you require many specifically crafted proteins to work together, and the probabilities easily get way beyond this. So yeah, it's generous, but I think that do you not see the relevance?
I do, yeah. I do. There's a couple people who have raised their hands. Well, why don't we start with Nate and then go to Nicholas? Feel free to ask your question.
Thanks. So I have a question about the cumulative case you presented, right? So this is kind of like a bit of a meta level question here. So this would include some of the evidence that you marshaled over the last 2 weeks. I'm imagining somebody saying, yeah, you know what, this evidence is extremely probative, and this is wildly disconfirming of the standard neo-Darwinian story. But they might then still wonder why it's confirming of intelligent design. Couldn't they just say something kind of like, I'm imagining something like Thomas Nagel's view, if you've read his Mind and Cosmos.
I have, yep. Mm-hmm.
Yeah, so the idea is something like, this doesn't support intelligent design. Actually, it just kind of leads us to rethink what we've been taking as like a certain set of assumptions, namely that like the fundamental laws, or maybe even just like the principles of biology themselves, like they're actually teleological in form rather than just purely mechanistic. We just need to work with a different conceptual scheme entirely, and we've just been wrong. What do you think about that kind of measure of resistance towards your argument?
Sure. So this is basically the question essentially boils down to, is this a God of the gaps argument? I think what if we're discounting the Darwinian story, then why does it follow that intelligent design is the correct explanation? And I think I addressed this to some extent in my first presentation, where I talked about the positive case for design, that when we look at the information systems in the cell, there's only one, the problem of the origins of life essentially boils down to the question of the origins of biological information. And all origins of life hurdles fall down uniformly, even if they solve some other problem or attempt to solve some other problem. They all fall down uniformly at the issue of the origins of biological information content. And biological information content is the one type of cause that, or one type of effect that is habitually associated with conscious activity as its cause. And so when we, I like to express things in terms of a Bayesian likelihood ratio. So what's the probability of this evidence given your hypothesis versus the probability of that same evidence given the falsehood of your hypothesis? And I think that the probability of encountering biological information by virtue of our uniform repeated experience is much more probable given design than given the falsehood of that, given the evidence that I've adduced on how difficult it is to get that biological information content. And furthermore, when it comes to irreducibly complex machines that we find in the cell that I discussed in particular in the first session, those require, it seems that those require a mind to assemble, because only a mind can visualize a complex end goal and bring things together necessary to realize or actualize that end goal. And also we talked about in one of the last session, I talked about the developmental trajectory, how you go from a zygote to daughter cells to 4, 8, and 16 and so forth. And at each stage, you're down regulating certain genes with respect to what their the future descendants of those cell lines are going to be doing. So that seems to require foresight. If you have to have an idea of where you're going, and so it seems that whatever process you invoke, it cannot be distinguishable from intelligence, because only intelligence has foresight. Does that make sense?
Yeah, it makes sense. And to be clear, I wasn't thinking of this as like a guard of the gap sort of charge. I often don't really know what those kinds of responses even amount to. Sometimes it's just like not liking inference to the best explanation. So I wasn't thinking of it like that. I was kind of thinking of it like this. So you rule out a certain hypothesis and your probabilities have to shift to some space. And I was just wondering why, particularly to the intelligent design hypothesis. And I think you did just answer my question. I guess what would really be interesting to consider is how people go about carrying out the Nagel-inspired project. Because on some of these approaches, you do have the supposition that like consciousness might be in some sense fundamental and not just local, but pervasive, right? So if you take like the David Chalmers inspired view, like something like a panpsychist view,
sure.
If you start off with that sort of thing, like consciousness is sort of like front loaded. And I don't know, I just wonder how much mileage people can get out of this to explain some of the local complexities that you cite and improbabilities.
Yeah, so that thesis would be consistent with intelligent design, wouldn't it?
Oh, I see. Nice.
Yeah,
Okay. Yeah.
So then the downstream question logically is, what is the identity of the designer? So the argument doesn't logically entail that theism is true. It doesn't, and far more so does it not entail that that Christianity is true. But it is evidence for theism, because I would argue that that the prior probability is higher for theism than it is for other competing explanations. And the basis for that would be the fact that we already have independent reason to think that theism is true, on the basis of the fact the universe had a beginning, which I think can be argued scientifically, and I think you can make even more compelling arguments philosophically as well. And also the fact the universe is fine-tuned, which seems to make more sense on design, and so forth. So I think that the prior probability of theism relative to other design candidates is higher.
Nice. Good. Thank you. Appreciate that.
Nicholas.
Yeah, good stuff. First off, I just want to, I agree with Finney. Appreciate both of you guys talking about kind of looking even-handedly at both sides of the argument. And for example, I like your discussion about the virology data. So I did have one question though. So when we're looking at, you had this string of characters argument. And also this kind of goes back to the Axe paper and the Nature paper. I'm curious what we're talking about when we say function.
What I mean when I say function. So a protein performing a function is it performs a certain role, such as catalyzing a chemical reaction in the cell, or by lowering the activation energy needed, or it contributes to some macromolecular machine. So what part of function do you find, or what part of what? I mean, I'm not quite sure what you're asking.
So I guess the problem here I have is that, like for example, in your string of characters argument, when we look at proteins, the majority of residues don't play a role in function. Like if you do mutational screens on a protein, you'll find a small portion of the protein that's involved in function. For example, like a catalytic triad or something that's involved in catalysis, and there's a small number of residues that are involved in that. Maybe you have some kind of allosteric that extends to other regions of the protein, but these are through certain like specific pathways. And so I wonder if it would be better to think about, to differentiate between protein function and protein foldability.
Yeah, so that's why Doug in his paper was looking at stability of protein structure. So does it form a stable structure?
Right. Okay.
Right.
Okay.
And being stable is generally a precondition for being functional, except in some cases where you have intrinsic disorder and so forth. But in most cases, being a stably folding protein is a precondition for protein function.
Right. Yeah. Yeah. I'm curious how he did this assay, because my experience has been is that frequently you can make mutations, a pretty good number of mutations that will maintain protein stability.
Yeah, it depends a lot on. I'll chime in here. So the protein that I studied when I was doing my PhD, it's called mTOR, or used to be called FRAP, FKB rapamycin associated protein. It's a huge protein. It's 2,549 amino acids long, and the protein that Axe studies was much smaller, 150 amino acids. And I think we have to acknowledge that the size of the protein is also very important here. So a lot of little proteins actually have less redundancy and wiggle room there, where a small number of mutations can just completely destroy it. In the case of mTOR, one mutation on the C terminus will eliminate the function of the protein. However, there's large stretches of the N terminus that do have more robustness for mutation. So I think your point is right, that we have to view this more as a gradient and a sliding scale there, given size of the protein, what domain we're looking at, and treat it not as a formula, but more as a principle. But I think the overarching point that is being made by Doug Axe there is that if we start with a small protein, something like 150 amino acid protein that he's looking at, it's hard to get to anything like functionality or utility there, whether it be structural or catalytic, in that range. And so, before we think about these.
So is he starting with just a random sequence?
No, it was an enzyme. What enzyme was it again?
It was beta-lactamase. It was beta-lactamase that confers penicillin resistance.
Right. And so what is he? So he's just screening through all possible mutations and looking at protein stability here?
So he actually modified 10 amino acid residues at a time, and the principle of his research is similar to the Nature paper in terms of its principle, and they come to similar conclusions about the rarity.
Right. There's other ways too you can do this. So there's an approach called CELEX, another Nobel Prize winning approach. Tomchuk and others.
We actually do CELEX in our lab occasionally.
Yeah, where you randomly make usually an RNA on a machine, and you just scramble up your ATCs and U's, and you can generate random sequences that way. And you can figure out, of course, that's RNA, not proteins, but you can figure out, ballpark how frequent it is to find something like, usually you get very, very weak function. I used to do CELEX when I was an undergrad. You get very, very weak activity in your first pass, and you've got to then take something that looks mildly promising and then do another round of CELEX on that, and often iterate multiple times before you can find something that is.
Right. Well, I guess one of the things I'm wondering is, how do we think about the probabilities if we have these negative and then neutral and positive mutations? So if we, start screening through mutations and anything that unfolds the protein, kills the organism, and then if it doesn't unfold the protein, it gets passed down as a neutral mutation. So we're scanning through, but there's dead ends for all the protein structure destroying mutations. And then the neutral mutations then expand throughout the population. So then how do we think about the computation of then screening these neutral mutations? And so we're maintaining the protein, the folded protein structure, and then maybe the going from one function to another within a folded structure is not so difficult.
Yeah, I think that's called stochastic tunneling in the literature. Basically using genetic drift or neutral evolution to get from one island of function to another. I don't think that I think that the probabilistic space is so vast that I don't think it's really going to help you to maneuver in that way.
Even with like the single-celled organisms?
Correct. Mm-hmm.
Right, and I want to do one more question here.
Sorry.
To close, but hey, that's great. I'm just going to build on what you said. Even if you have a single cell organism, you've got, say, a promising mutation over here, and then you've got another organism that's got a promising mutation over here, right? Then you've got to figure out, okay, well, how likely are they to recombine? And there's this notion of search space and search trajectory, right? Which is kind of adds even more complexity there, even if you do assume something like recombination and meiosis that can potentially bring them together. So it's identifying the right path to go from very small or nothing to actually build up a full length functional protein. Presumably that's not going to come from just one lineage that's just finding it there. It's got a little bit over here, a little bit over there, and you're multiplying out. So the probabilities do get very small. Last question here before we break, and then as Jonathan said, you can reach out to him if you have further questions and want to dialogue. So the question is around, he says, what really resonates here is both sides need to be honest in order to have dialogue. It also reflects the current political discourse. That's true. Unwillingness on both sides to concede any points, no matter how valid they are, which results in both sides talking past each other. I'm curious if we can go back to Jonathan's early argument against hotspots for preferential insertion of viruses. I agree that this observation is one of the most compelling arguments for common descent, but I didn't understand your argument against the hotspot response, i.e. viruses could have independently inserted into similar regions across distinct species. If you have any resources that you think are helpful on this topic, please share. Thanks.
Yeah, so basically I was saying that there is some level of target site preference, but it doesn't seem to be anywhere near sufficiently locus specific in order to be probabilistically helpful here. That it would need to be much more locus specific or site specific integration. I hope that clarifies that.
In other words, you're saying like the virus will prefer, I'll just make something up, an AGG-AT, and there's 100 of those. Again, I'm making up numbers here. There's 100 of those in a genome, but instead what you find is that the virus is every time it's right upstream of dystrophin in a variety of organisms, which is one of the 1,000 places that it could have integrated in.
Yeah,
so it seems like the sequence specificity is not. There's more to it than that. Like it seems like there was an actual event that happened upstream.
Exactly. So it has a target site preference for CPG islands, I think, and close to promoters. But there's so many of those, right, that it's not really probabilistically helpful. So another interesting observation, by the way, on this is that often critics of intelligent design will criticize the concept of specified complexity that I talked about in the first talk, and they'll say that specified complexity is an incoherent concept. Geoffrey Shellett and Wesley Ellsbury, for example, have tried to make this argument. They're mathematicians, and the problem there, though, is that virtually all of at least the molecular arguments for common descent are based on arguments from specified complexity, right? That any distribution of point mutations or any distribution of retroviral inserts or what have you are equally improbable as any other, right? But there's conformity to some independently given pattern, which is a signal of common descent. That's the argument. So in the process of making the argument for common descent, they are basically granting the specified complexity is a legitimate concept. So that's an interesting irony there as well. And specified complexity, I would argue, can be transmitted by two different routes. One would be that it's instantiated. Can you
explain specified complexity?
Yeah. So specified complexity is where you have not only improbability, and improbability would be, and to qualify as being improbable in this sense, it would have to exhaust available probabilistic resources at one's disposal, the number of opportunities you have to get it right. Specification would be conformity to some independently given pattern. So the statement that in the beginning was the word, for example, the first verse of John's Gospel, is independently specified because it communicates meaning. It has a semiotic dimension, and it's also incredibly improbable. If you're just typing random letters on a keyboard, it's very unlikely that you'll stumble upon that by chance. So that would be specified complexity.
Got it. All right. Well, why don't we wrap up for today? So just to give everyone a little bit of of a tour here. So first, thanks to Jonathan for his giving these 3 sessions there. So thanks for that. We're gonna move on to a different topic. For next time, we have Chuck Pike next week, and then Dick Keyes the following week. So some of you have heard both of them. They spoke here. Chuck Pike, his specialty is Bible prophecy, and Dick Keyes is with an organization called Labrie, and they do all kinds of apologetics. So I'm excited for that. I'll send out an email. Hope you can join there, and yeah, look forward to seeing you all.
Vinny,
next
can
time.
I say just one comment
Yeah,
here at the
please.
end?
Yeah,
So
please.
we've recorded these 3 sessions, and it's on the cloud and it's totally accessible. What's interesting about it is that also the chat is recorded, and also there's a transcription of the lectures, and so all that's available online, and we're glad to share the links for you.
Yeah, the links have been in the bottom of the emails that I've sent out, so you can listen to the previous ones in case you didn't catch everything or want to go back there. So, and I actually put another YouTube video that I recommend. It's a dialogue between Stephen Meyer and David Berlinski that covers from a slightly different perspective some of the things that we heard today.
So thank you, Jonathan. Thank you, Finney.
Thank you.
Yeah, you're welcome. Okay. Bye-bye, everyone.
Bye.